Uh, Today we will talk about the replication of DNA. DNA consists of a phosphorus, a deoxyribose sugar, and a nitrogenous base. It has a double helix with a phosphodiester and a hydrogen bond. DNA makes up the genetic code. The process of DNA replication for eukaryotes is the following. First step, DNA helicase unwinds and unzips the DNA parental molecule. 2. DNA polymerase creates complementary strands, and the replication is always semi-conservative. The parent strand is paired with a complementary strand, and the DNA polymerase finds free nucleotides and pairs them with the correct base on the parent strand, creating the complementary strand. And A always pairs with T, and G always pairs with C. So.
Did it save?
So you have to talk now.
Oh, it's just paused. Okay. Oh no, it's still going.
Yeah, because we keep going.
Okay. Within the DNA helicase works fully on the leading strand, which is 5' to 3'. On the lagging strand, which is 3' to 5', the RNA primer is used as an initiation site. On the lagging strand, Okazaki fragments are created due to the DNA polymerase III skipping parts on the parent strand due to the 5' to 3' sequence. The DNA ligase attaches the Okazaki fragments together. DNA polymerase acts on the 3' OH of the existing strand for adding free nucleotides. Comparison with the replication of DNA between eukaryotes and prokaryotes. Prokaryotes contain DNA gyrase, which relieves the strain while the DNA is being unwound. It is also called topoisomerase. Replication is in a bidirectional and goes around in a loop. Some important terms include semi-conservative, which means there is one old strand and one new strand from the original DNA. Helicase, which unwinds the DNA and breaks its bonds by the pairs. And DNA polymerase III, which attaches nucleotides in a 5' to 3' direction.
DNA ligase attaches the Okazaki fragments together. The Okazaki fragments are sections of new DNA on the lag strand, and the RNA primers are the initiation site for the DNA polymerase III on the lag strand. So thank you guys for listening, and hopefully this podcast on DNA replication has helped you.